Hi, everyone. Good morning. Well, not good morning more. It's good afternoon over on the East Coast. But my name is Lisa Truong. I'd like to thank the organizers for inviting me to give this update on the toxicity testing we've been doing in zebrafish. And so with that, I wanted to just acknowledge that I'm at Oregon State University and I'm in Dr. Robin Tangway's lab and representing some of her work that we've been doing in collaboration. So just kind of a quick outline of explaining kind of why we're using zebrafish, some of the botanicals we're testing in this the data we're showing, and some of the results and summary and the future direction. So we've talked a lot about the different model systems. Thanks for the introduction from everyone else. I don't have to talk too much about it, but we're trying to develop models to actually take advantage of the advances science is having. And so we want to use models for the fit for purpose and don't want to overlap and establish methods that can't be used for understanding how to design safer alternatives or identifying safer alternatives. And we want to be able to understand the biological consequences before the adverse effects happen. And so with that, I just want to I'm not sure how many people know, but why using zebrafish? And one of the main reasons is that the zebrafish has a lot of the organ systems are very similar to humans. They have a central nervous system, they have a cardiovascular system, the liver is there, a gallbladder, pancreas, and intestine, and the advantage of being smaller. And this is an in vivo system, which makes it comparable at the genomic level to like the humans, the mouse, chicken. And you can see here on the Venn diagram that in the center, there's about 10,600 roughly genes that are conserved across all the models, which make it biologically relevant. So the approach that we have in the lab is that we're using a systems approach to actually identify what could be what a toxicant, a chemical, or botanicals in this instance can be causing an effect. So the molecular signaling is conserved in humans, like I just mentioned, but there's also 80% homology with humans, where most human diseases can be modeled in zebrafish. We can leverage the technical advances that have been previously talked about of cell culture and imaging, and also being an in vivo system at the same time. One interesting thing about zebrafish is that at 72 hours, they're metabolically capable. So we can look at before the metabolism gets done to affect with their liver. And we can actually do whole animal mechanistic studies. So what we like it to do is looking at using chemicals, expose them to zebrafish and figure out what genomic response is happening that leads to a phenotypic response. So today I'll be kind of presenting the data for 16 of the full 16 botanicals we talked about earlier. I'm not going to read through all of it, but we did a lot of different concentration ranges, and I'll go through that in a minute here. But just kind of overview of what the toxicity pathways / workflow that we do is, where essentially it's a bioactivity screen. We take zebrafish embryos, we expose them in the water in their 96 well plates, and then we have 2 behavior assays that I'll be talking about. So as you can see here, embryos are collected between 0 to 4 hours, and this is just an image of what looks like. We have these large tanks that have about 100 gallons and holds about 300 fish, and we can basically set them up the night before, which means we put a cone in, and with zebrafish, once the light turns on, the females will lay their eggs, and the eggs are externally fertilized, and then we have them collected into sediment filter, and we can kind of turn the spigot and collect them in the strainer that used to be there, and we wash them into a petri dish. Afterwards, you can see at 6 hours post-fertilization, right before that, their embryos have kind of developed from a single cell to about a couple thousand cells. And at this point, we actually remove their chorions, which is kind of an envelope, that like eggshell around them that protects them. We do this to ensure that there's no barrier to actually the chemical contact or botanical contact instance to understand what is impacting. And so what we do is then deliver one embryo at a time into a 96-well plate. And we used to do this back for a while by hand, but in the past, we have been, in 2010, we developed these robots, instruments, where we can actually pick a single embryo up and put them in the 96-well plate over time. And you can see, what you see there is an image camera above that can actually find the pickable targets and provide it to the robotic arm to put them into 96-well plates. Each well has 100 microliters in there, and we put the embryos in and then add chemical. And one way we add chemical is actually using a digital dispenser, which means we don't have to worry about cross-contamination or a graduate student or technician doing calculations off. We use something called the HP digital dispenser, which is where you can see these little reservoirs. You add the chemical in there at stock concentration, and using the inkjet technology that HP developed back in the day, they adapted it so that they can precisely dispense nanoliter to picoliter droplets to achieve that desired concentration. They have a really nice software, and you just put a plate in, and they'll do the work for you. So that kind of brings us to how we accept the exposure. And this all happens within the first 6 to 8 hours, which is prior to organogenesis. And so we ensure prevent evaporation, we seal up the plate, and we put them into the incubator. And at 18 hours later, which is their 24 hours post-fertilization, we do a behavioral test. At this time point, they do not have eyes yet, but you can see that they have grown from a circular ball of cells to now having a tail extended, and they have a yolk sac that they're using, but their eyes are not functioning yet. So we have this photomotor response assay where we keep them in the dark the whole time. We don't know most of the time we actually test in chemicals. We don't know their liabilities, so we prevent that by having it covered in aluminum foil, and then we put them in this assay instrument where they're in the dark for 30 seconds. Then we shine a pulse of light, and then we let them habituate a little bit, and then 9 seconds later we hit them under the pulse of light. This assay works on the photomotor receptor cells, which is in their hindbrain, and at this point, at this life stage, you can see that there's tails, so they start contracting, which is a involuntary action. This is natural for them, and with that assay, we're able to detect if they, when you shine a pulse of light on them, you see this excitation, which is normal, and then if you shine another pulse of light, the cells have already been excited, so they won't recharge, and so there will be less movement. So we then afterwards put them back into the dark in incubator, which is at 28 degrees Celsius, and we leave them until 120 hours post-fertilization. At this point, they are they're a free-swimming animal. They have all the organ systems, and we can start then putting them into instruments such as, turn off the laser pointer for a second. So we have 9 of these viewpoint behavior assays viewpoint zebra boxes that you can actually use to track the movement of the fish. And like I mentioned, they're free swimming fish at this point. So what we take advantage of is that at this life stage, their eyes are functioning and they can respond to light. So we turn the lights on and off on them. At the typical fish and the light, they will freeze. And in the dark, when you have the lights off, they will start moving around more. And so we have a 26-minute assay where we turn the lights on and off every 3 minutes, and you kind of see this behavior where there's not much movement. There's not much movement, and then there's movement, and then it cycles back and forth. Then after we do this, we track their behavior, we can actually assess if they're phenotypically normal. This is the screen we have is using visual machine learning in addition to actually having human assessments of the embryos to see if there's any malformations, which you can see any abnormalities in their in the fish at this life stage after exposing continuous static exposure from 6 hours post-fertilization all the way to 5 days. And this is using a hormonal optics, which is a pretty interesting and amazing camera system, microscope system, where you can put a 96 well plate and it has 52 sensors that can take an instant image of all 96 wells at a high enough resolution for us. So now to get into that, now that that's a overview of what our assay looks like, I can kind of go into our experimental design. So for these botanicals, we started off with doing a range finding test, which means we wanted to see do a limit test to see what at the low, what malformations we would see and what the high would be up to a reasonable solubility. So we have a set standard concentration of 0.78 micrograms per mil, all the way to 250 micrograms per mil. These chemicals were dissolved in DMSO when possible, when they were provided to us in powders. And the reason we need DMSO is because of the digital dispensing I mentioned earlier. They need a solvent to allow for that, and we normalize the 96 well plate so that there's no plate of chemical effect. So we have the 12 concentrations that we test, and then we follow up with a definitive testing, which is a secondary test where we focus in to try to create a nice dose response curve if possible. So at this point, we would have 3 replicate plates. You can see the design here where the first row is all controls, and we have multiple concentrations of each of the chemical across 3 replicate plates. The reason for more concentrations versus replicate animals is that allows us to be able to identify the points of inflection for future studies where we're looking at trying to identify benchmark dose. And so we have 13 morphological endpoints, and we have 2 behavioral tests as I described earlier. So kind of jumping into the results a little bit, where you can see here on the right is a heat map. On the y-axis is the extracts themselves. Along the x are the different endpoints that I was talking about. So the first set here, you can see is where we looked at morphological effects. And then we have a summary effect called any effect of any, where we just kind of use Zebrafish as a biosensor, where we can say this chemical was there and there's something wrong. Not necessarily reading into exactly what is wrong, but at least it alerts us to helping hazard identification. Then EPR, which is our embryonic photomotor response at 24 hours, I was mentioning, is here. And then we have the 5-day, which is behavior, which is the LPR. And the colors are on 2 different scales because of the range. Unfortunately, this is kind of more preliminary, where it's the different ranges because of some of the chemicals were more potent than others. But what you can see here is that we have milk thistle and ashwagandha and Houston extracts are the only ones that did not cause any morphological effects here, which which are the white cells here. The darker the color is, the more potent it is. And so then we also have 2 chemicals that also only cause morphological effects, but did not cause any behavior effects. So another way to look at all this data is to actually dive into each individual endpoint. And as I mentioned before, we tested them at 12 different concentrations. And so these are stack plots where you can imagine that every circle represents an individual animal. So at a certain point with statistically significant threshold of 0.01 using Fisher's exact test, you can determine within one endpoint compared to the control if there was a significant difference, and then it will turn red. So the best way to interpret that is any of the red dots would mean that the animal was there was a statistically significant. So the first 3 endpoints are evaluated at 24 hours, which is 18 hours 18 hours of exposure time, and mortality is observed. This more endpoint onward is the endpoints observed at 5 days. So you can see that, for example, the blue kosh has malformation, has mortality observed, nothing happening at 24 hours. There's no mortality, immediate mortality, but by 5 days, there is a dose response, dose-dependent increase in mortality observed. And one of the reasons we used the any effect as our summary endpoint is because, as you can imagine, as there's higher concentrations, there's more mortality observed. The denominator of how many viable animals to evaluate for morphological and abnormalities is diminishing. And we don't do any malformation evaluations on animals that are dead. And so you can see here that the ephedra at 24 hours already has this nice dose response curve you see, and by 5 days on, there's a couple more malformations, but not a large increase. And you can see that on the right, some are more potent than others based off of the lowest effect level. And so you can see there's milk thistle had no statistically significant morphological effects. So what you see here all next then is also aswana, what I mentioned earlier, and then Eucina, where you have at the highest 2 concentrations. And so some couple more chemicals that just shows another way. And what the goal was to, like I mentioned, the definitive testing is to try to achieve a dose response curve. And this is just some examples of what we were able to do with the zebrafish model, where we can, with the 12 concentrations, fit it to a pretty nice curve. Some are better than these. The Thunder God is better than the others, but you can see that there's a nice dose response curve where we can predict a BMD10, an EC or EC50 and EC80, which we can use later on for transcriptomics. So we also, like I mentioned before, the embryonic photomotor response at 24 hours, where the first 30 seconds is in the dark, which we call as a background phase. When we hit it with this, a pulse of light, it excites the photomotor cells in the hindbrain, and you expect this excitation, which you see in the black, which is the control. And then when you hit a pulse again, there's no behavior. And for all the chemicals, all the botanicals that we've tested, there were 13 that caused an abnormality in this effect, which typically meant they had less at the higher. There's a dose response, dose-dependent response, where the higher concentrations had less and less movement. And just to note that we do remove the animals that were already dead, so those do not attribute to this a flat line or hypoactivity. And 3 chemicals in particular cause an abnormal behavior that they affected all three phases, one that typically isn't very typical, which is aconite, comfrey, and eucina. And just as a reminder, we also did the larval photomotor response, which is when they're free swimming, and you can see this behavior. At this point, the animals that are malformed or dead are removed, and at concentrations where if there was more than 30% of the animals were malformed, they were not included in the statistical analysis to make sure that we are looking at behavior effects, not systemic toxicity. And from this, you can see we split it out for Uhimbi, where at the controls, you can see that in the light, they weren't moving very much, and there's this peak of movement. And these are the 7.8 micrograms per mil. You can see that this peak doesn't happen, and they're causing this abnormal swimming behavior. We can't determine or attribute if the behavior is due to the vision or if it was muscle. We just know that there is a decrease in ability to move, respond to the light. More studies are needed to actually determine what the mechanism is. And so, kind of to bring you back to the summary of the heat map again, what you see here is the ephedra, which causes only developmental toxicity. There is no behavior effects or whatnot. And then we have 10 out of 16 that causes developmental neurotoxicity, which means they cause more malformations, and it had effects in the cause abnormal behavior in any of the assays. And there's 3 chemicals that cause no which is listed here in the green, that had no developmental abnormalities, but they had behavior effects. So kind of a conclusion just at this point is where we're able to profile 16 the botanicals in the Zebrafish model and high throughput platform. We are able to identify early life stage behavior functions that are sensitive to this short period of exposure, and that static exposure. Milk thistle and ashwagandha did not cause mortality or malformations, but are associated with abnormal photomotor response and the startle behavior, which I didn't have enough time to talk about. And then this is just kind of a summary that we're able to use the power of Zebrafish to identify developmental and developmental neurotoxicity in these chemicals, and kind of future directions. I took the data and kind of able to show the dose response of the any effect, which is a summary endpoint that we talked about at 5 days. And here on the X is the concentrations and the 16 botanicals, and you can see there's a difference by the different colors of the potency. And so with this information, what we're hoping to do is be able to conduct global transcriptomics at multiple concentrations. We're doing 5 concentrations to start to elucidate mode of action prior to the onset of adverse effects, which means we're going to look at them not at 120 hours when we see the malformations or the behavior effects. We're actually going to look up prior to that with 48 hours at 5 different concentrations at the EC10, 50, 70, 80, and 100, I believe. And then we're going to start to be able to look at that data to kind of see what could be going off, be manipulated or affected earlier and to result in abnormal endpoints that we're seeing. And then we might be able to start comparing results from other complementary test platforms that we've been hearing before today. And with that, I'll take any questions.
Great. Thanks so much, Lisa. That was a really excellent presentation. If folks have any questions, they can put it in the chat or raise hand. I do see one already from Gertrude. They stated that the zebrafish is often used as a model to investigate the impact of chemicals on pigmentation, as some may stimulate while others may inhibit the melanin synthesis. Is that impact of pigmentation of interest regarding the botanicals? So I can answer that part. We didn't specifically ask to look for it, but maybe Lisa, you could speak to any of your work if you've used the model to look for that effect.
Yeah, so we can do that. It's just more where zebrafish is transparent up to the first about day or so, and we actually have chemicals. We know certain chemicals can remove the melanin synthesis, like you mentioned. So we could have done it, but we didn't, like you said, Connie. That makes it challenging.
Yeah. Have you done that for any other chemicals, Lisa? Is that something clients or like your own research have done? Yeah.
Yeah, so we can actually tell there's some compounds actually cause them to be hyperpigmented versus some that should remove the pigmentation. Robin has a grant where she's screening about 6,000 chemicals that she is working in collaboration with the US EPA CCTE, and we do look at chemicals to look for pigmentation.
Thanks for that. As we're waiting for questions, one thing that's come up in a lot of discussions is the thoughts on, are the chemicals actually getting in? And with complex mixtures, that ends up being, a whole nother layer, because it's how do we measure? Could you speak to maybe what your lab have done with single chemicals to look at the actual uptake into the system?
So we have been working with, we have a Superfund Research Center, and we've been working with Pacific Northwest National Laboratory to actually measure the amount of compounds of pHs, in particular polycyclic aromatic hydrocarbons, and actually being uptaken in the fish, how much is absorbed to the plate, and actually how much is being volatilized. And so we're starting to take a more mass balanced approach to develop models to figure out what parameters can be used to predict, and that's being applied to mixtures and pHs. So I can see that in the future we're going to be able to apply to mixtures, such as actually synthesizing the mixtures and knowing, okay, these are the properties individually, and look at additive or synergistic effects.
Yeah, that's something we've thought about before. And then for every model system, it's wondering, how could we actually trace what goes through giving this, mixtures problem, which is what this all is. So like, I think leveraging learning from PAHs and other types is really important. There was one question. Has your lab looked at essential oils?
Yeah, actually, we are actually starting to look more at essential oils. We have a grant with the USDA to actually look at essential oils and their ability to be more active as a sprout inhibitor for pesticides, instead of pesticides for potatoes. So we're actually starting to dive into more essential oils, and we actually found that there's um, they're less toxic, but there are some less toxic, and we have collaborators actually synthesizing mixtures and actually identifying what the components of them are.
Did you all have to adapt your setup based on, just their differences in maybe absorption or solubility?
So we haven't had to, but we're actually working with them to figure out how we can measure that the metabolites and everything in the fish to see that. But we are like fortunately our digital dispenser can dispense essential oils without adding this extra surfactant into it or solvent to dissolve it. But most of them at high concentration start being an oil, so there's that effect.
Great, thanks for that. Olaf had a question. To which extent can behavioral and neurotoxic effects be differentiated?
That is a question where we're working on the whole field is kind of working on, but we can, the nice thing about the model is that we can manipulate when we start the exposures. So if we can actually separate out when to add the chemicals, if it's after, central nervous system has been developed, we can actually expose them at that point and actually determine if it's neurotoxic versus actually then looking at, okay, if those genes are knocking out certain genes or then also washing out the solution. So prior to the behavior assays, then you can actually start assessing what genes are actually affects their being and also looking at the genes prior from the transcriptomics and that should give us some insight.